Hi, so my talk is about the importance of including prey in assessments of stressor impacts on top predators. So with lots of concern about the amount of human activity going on in the marine environment, there's concern about how the impacts on various top predators, considering the harbour porpoise here. And the main consideration here is about how disturbance may have knock-on impacts on the foraging of animals, which can have effects on the animals' energetics, and therefore their chances of survival and breeding. So here I assess the importance of including different prey types of size and different qualities combined with foraging data collected on live wild harbour porpoises in order to better understand the possible energetic consequences of disturbance and what those knock-on effects might be. The key things about this study is that it really highlights the importance of further research into predators' prey and the wider environment in order to better understand the ecology and imbalance of the ecosystem.
When talking about multiple stressors, we often discuss them in terms of addition, synergism, and antagonism. But are we always using these terms correctly? The awkward truth is that we probably aren't. Join me on Tuesday morning in the multiple marine stressors session, where I'll describe where people go wrong with the reporting of their data and the consequences that this can have when interpreting results within the literature. But more importantly, I'll discuss why people continue to use these terms incorrectly, why doing so not only makes a lot of sense in some situations, but may actually be an option well worth exploring.
We've all seen and admired views like this, but have we ever considered the effect of artificial light at night on the environment? My name is Eleni Christoforou. I'm a PhD student at the University of Glasgow, where I study the effect of artificial light at night on mussel activity response. As part of my experiment, I exposed mussels to 3 artificial light at night treatments and a controlled dark treatment. Throughout the day, the mussels are exposed to continuous daylight illumination. Throughout the 24 hours, I tracked their gaping activity. You can see a mussel opening from a closed position, equipped with a recording device. See you at the conference for more details and preliminary results.
Hi, I'm Glenn. I'm giving a talk on the use of biomarkers in relation to climate change at this year's MASTS annual science meeting. In short, the temperature of the upper 75 meters of the ocean has been increasing and the pH has been decreasing for decades. This might continue depending on human activity. In addition, local fluctuations in salinity might become more extreme due to increased precipitation and evaporation. A biomarker can be defined as an objectively and quantitatively measurable response to exposure. One example could be by measuring DNA strand breaks, as in the work I will be presenting. The aim of this research is to probe how biomarker responses may be impacted in the future. To do this, I expose indicator organisms to selected future conditions in the marine environment in relation to climate change and study established biomarkers. Early results of my work indicate that either an increase in temperature or extreme salinity fluctuations cause an increase in DNA strand breaks in the gill tissue of mussels. When the stresses are combined, this response can also be observed in other tissue types.
Hello, my name is James Rimmer, a PhD student at the University of St Andrews in the Sediment Ecology Research Group, and I'll be talking in the Multiple Marine Stressors Forum on the 6th of October about the cumulative impacts of multiple marine stressors on depositional estuarine habitats. I'll be talking about some of my active areas of research, the methods we can use to quantify impacts, and how both mesocosm systems and fieldwork experiments can be used to understand the interactions between multiple stressors.